Good morning to all. Today I am going to deal with the topic anatomy of hypocotyl. So let's start. First of all, I'm going to explain about the seedling, its different parts, and how seedling emerges, etc. Seedling is a young sporophyte developing out of a plant embryo from a seed. And seedling development starts with the germination of a seed. A typical seedling usually consists of three parts, that is radicle, hypocotyl and cotyledon. Radicle is also known as embryonic root, hypocotyl is known as embryonic shoot and cotyledon also known as seed leaves. We all know that some seedlings has no cotyledon at all, and such seedlings are known as ecotyledons. And based on the number of cotyledons, there are two types, monocotyledons and dicotyledons, and the meaning of the term is known from the very word itself, that is monocotyledons possess only one cotyledon and dicotyledon possess two cotyledons. And earlier we have said that a typical seedling consists of three parts, that is radicle, hypocotyl and cotyledon. And this radicle give rise to root and hypocotyl to the lower stem and the epicotyl to the leaves and the upper part. That is, first of all the radicle emerges which give rise to true roots and which later grows as an important organ or for absorption or anchorage into soil, and later hypocotyl emerges and produce the lower stem and later epicotyl to the leaves and the upper part. And now let's get into the topic, that is anatomy of hypocotyl. First of all, let me explain about hypocotyl, its emergence and etc. Hypocotyl is the stem of the germinating seedling found below the cotyledon and above the radicle. That is, the position of hypocotyl is below cotyledon and above the radicle. In this picture, we can see the position of hypocotyl, that is, it lies below the cotyledon and above the radicle. Later, this hypocotyl, after the hypocotyl development, it gives rise to the development of true leaves, that is, plumule gives rise to true roots development. As the plant embryo grows, it sends out a shoot called radicle that becomes the primary root, which then penetrates down into the soil. And this radicle which produces the primary root is important because through roots, absorption, anchorage and many other process which is vital for a plant takes place. So radicle development is very important in case of a seedling. After the emergence of radicle, hypocotyl emerges and it left the growing tip including the seed coat above the ground, and later plumule give rise to first true roots. That is, first of all the radicle develops which give rise to roots, and later the hypocotyl develop and it lift the growing tip of the plant or the seedling including the seed coat above the ground, and later this plumule produce true leaves. And this germination of a seedling is explained or clearly seen in this diagram. That is, first a seedling produce radicle, later hypocotyl is produced, and we can see the position of cotyledon, radicle, etc. in this diagram. And hypocotyl is actually considered as the primary organ of extension of young plant. And in some plants, hypocotyl act as a storage organ. That is, in cyclamen, this hypocotyl act as a storage organ and it is known as tuber in case of cyclamen. It is based on the growth of hypocotyl elongation, the growth promoting and growth repressing effect of the plant hormone ethylene is studied. That is, under normal light condition, hypocotyl growth is controlled by a process called photomorphogenesis. That is, under normal condition, hypocotyl grows properly. That is, it does not shows any growth repressing effect or extra growth. That is, it grows in a controlled way. And when shading evolves, a negative photomorphogenesis occurs. As a result, increased growth rate of hypocotyl occurs. So, with this we understood that the rate of hypocotyl is highest in case of darkness. And in this diagram, we can see seed coat, cotyledon, hypocotyl and radicle. This radicle first produce root, hypocotyl produce the stem, and later plumule give rise to first true root, true leaves. And now we can detailedly explain the anatomy of hypocotyl by using the example country bean, that is Lublub purpuris. It is a scientific name of country bean. The basal part of the hypocotyl is root-like, while the middle and upper part are stem-like. That is, this is the first important feature regarding the anatomy of hypocotyl. The basal part of the hypocotyl is seen as root, and the middle and the upper part are stem-like. And we can see a transition of vascular tissues that occurs in the basal part of hypocotyl, and vascular bundles are usually arranged in collateral in the middle and upper part, and towards the basal part it is arranged in radial fashion. And we can see two types of vascular bundles, that is small and large vascular bundles, and one can see one or two small vascular bundles in between two large bundles. And this large vascular bundles contain xylem and phloem, and small vascular bundles may or may not contain xylem and phloem. This is a diagram of the vascular bundle arrangement. In this we can see a small vascular bundles in between two large vascular bundles, and we can also see the arrangements of vascular bundles, that is collateral in the middle and the upper part, while radial in the basal part. Several poles of primary phloem are seen outside the primary xylem in the anatomy of this plant. And in the basal part of hypocotyl, 4 strands of xylem is seen alternating with the phloem strand, and each xylem strand is divided into 2 xylem poles which are either connected or separated. That is, in the basal part of the hypocotyl, 4 xylem strands can be seen, and these xylem strands are usually arranged alternating with the phloem strand. And this xylem strand is divided into 2 xylem poles which are either connected or separated. And this is actually the diagram of the anatomy of hypocotyl of country bean, and it's not clear. I hope it's visible. And the vessels in the xylem strands are usually radially arranged, and we can see a lot of tanniferous cells with or without content in the primary phloem region. That is, a large amount of tanniferous cells, some with content and some without content, has been seen in the primary phloem region. And the arrangement of pericycle is discontinuous in the anatomy of hypocotyl. And two adjacent grooves of sclerenchyma are connected by 1 or 2 layers of sclerenchyma cells, and cambium can also be seen, and cambium appears and become active in the fascicular region earlier than interfascicular region. That is, cambium become active in the fascicular region first when compared with the interfascicular region. And in the basal part of hypocotyl, the cambium of 4-day-old seedling gradually extend towards the upper part. That is, in a 4-day-old seedling, we can see the cambium gradually extending towards the upper part, and when we have a look into the 7-day-old seedling, the cambium usually forms a ring. And this anatomy of hypocotyl shows epidermis, cortex and pith that resembles a typical dicot plant, and phellogen appears in the cortex and give rise to cork and phallodem, and we can also see periderm in the anatomy of hypocotyl of matured plant. So this is all about the anatomy of hypocotyl in case of country bean Lublub purpuris, and after this we can discuss about cottonseed. That is, here we are explaining the anatomy of hypocotyl by using two examples, that is country bean, that is Lublub purpuris, and in case of cottonseed, first we have discussed about Lublub purpuris, that is country bean, and now we are discussing about the cottonseed. In case of cottonseed, the hypocotyl is well developed and stem the hypocotyl is well developed the stem between cotyledon and radicle. And transition of hypocotyl can be seen from hypocotyl and radicle. That is, we can see a transition from hypocotyl and radicle in case of cottonseed. That is, the tetrarch arrangement of typical root vascular system changes to a circular stele of typical aerial stem. And we can see a tetrarch arrangement changes to a circular stele. And this is the diagram of the anatomy of hypocotyl of cotton seed. In this we can see this is the chelacal end and this is the micropyle, and a radicle is placed here, and next come hypocotyl, epicotyl and cotyledon. And upon germination, water enters through chelacal pore and it travels towards the radicle, and after the absorption of water, this radicle elongates and it breaks through micropyle of seed coat and becomes seedling root. That is, during germination type, water enters through this chelaza, and this water reaches this radicle, and this radicle elongates and comes out through this micropyle. That is explained here. That is, water enters through chelacal pore, travels to radicle, which elongates and breaks through micropyle of seed coat and becomes seedling root. And water entering this root hydrates the hypocotyl, which elongate and push the cotyledon through the soil. And this hypocotyl forms a hook when it penetrates through the soil, and this hook is the first tissue to emerge from soil. And after emergence, hypocotyl straightens, allowing the cotyledon to function as photosynthetic leaves. This is how germination occurs in the seedling. That is, first when water comes through chelaza, radicle absorbs it and it elongates and breaks through the micropyle, and later hypocotyl emerges, and this hypocotyl produce a hook as it penetrates the soil, and this hook is the first tissue to emerge from the soil, and after emergence hypocotyl straightens, allowing the cotyledon to function as photosynthetic leaves. That's all about germination. And let me conclude now. And in this slide we have discussed first of all about a seedling. It's three parts, that is radicle, hypocotyl and cotyledon. And next we discuss about hypocotyl, that is it is a stem of germinating seedling found below the cotyledon and above the radicle. And first of all, radicle emerges and produce true roots, and later hypocotyl emerges and it lift at the growing tip, including the seed coat above the ground, and later plumule give rise to first true roots. And this hypocotyl also act as storage organ, and sometimes they are known as tubers in cyclamen. And here we have explained the anatomy of hypocotyl by using the example country bean and cotton, and we can see the basal part of hypocotyl as root-like and the upper part stem-like, and vascular bundles are collateral in the middle and upper part and radial in the basal part. Two types of vascular bundles are also seen, that is small and large vascular bundles, and they are arranged in between two large bundles. And several poles of primary phloem outside primary xylem is present, and we can see four strands of xylem alternating with the phloem, and this xylem strand is divided into two xylem poles which are either connected or separated, and lot of tanniferous cells with or without content has been seen, and pericycle is discontinuous, and we can see cambial growth. And in a 4-day-old seedling, the cambium gradually extends towards the upper part, and in 7-day-old seedling, this cambium usually forms a ring. And this anatomy of hypocotyl resembles a typical dicot plant having epidermis, cortex and pith, and hypocotyl also possesses phellogen, periderm, etc. And in case of cotton seed, hypocotyl is a well-developed stem between cotyledon and radicle. Transition from hypocotyl and radicle can also be seen in this cotton seed. And next come the germination. And that's all. Thank you.